So welcome all, and thank you for joining us today for, as Natalie's just said, a first in what is a series of webinars co-hosted by Outer Sciences and Signature Discovery around the theme of CNS drug development, and in particular CNS safety assessment. So that we'll be addressing this topic from a translational angle, bringing together non-clinical and clinical insights into what we see as some of the key aspects of CNS drug development. So today's webinar, the first in this series on the topic of abuse potential, is titled To Control or Be Controlled, Navigating the Abuse Potential Evaluation of CNS-Active Drugs for EU and US submissions. Can I have the next slide, please? So just some quick introductions to kick us off. My name's Andy Mead. I'm Head of Drug Abuse and Substance Use Disorders at Signature Discovery.
Hello, thanks Andy. And I'm Beatrice Setnick. I'm the Chief Scientific Officer at Alta Sciences. Next slide, please. In today's session, we will explain why abuse potential is evaluated and how this impacts the scheduling of a drug once marketed. We will also discuss the key differences for evaluating abuse potential for EMA versus FDA drug submissions. You will learn how to determine if your CNS-active drug requires abuse potential evaluation, and if so, what are the required preclinical and clinical studies, and when is the best time to conduct these studies in your drug development program. Lastly, we will provide insight on strategy and discuss situations where waivers may be considered. The session will conclude with an opportunity for our audience to ask questions. Next slide, please. CNS-active drugs have unique attributes that require specific evaluation during the drug development process. One must consider any cognitive impairing or enhancing effects, the potential of a drug to cause physical dependence and withdrawal, and the potential to cause additive effects on impairment when combined with other drugs or alcohol. All of these topics will be covered in our future webinar series. Today, we will be focusing on the reinforcing and psychoactive effects or abuse potential evaluation of CNS-active drugs. Next slide, please. First, let's discuss the varying terms that are used in this field. The FDA uses the term abuse potential, whereas the EMA uses the term dependence potential. Despite the different nomenclature, both terms relate to the likelihood that a drug has addictive properties and may become abused. This is in contrast to the term physical dependence, which is a distinct term referring to the physical adaptation that occurs during chronic drug exposure. This may lead to withdrawal syndrome when a drug is suddenly discontinued. Physical dependence evaluation is an important part of the abuse potential evaluation, and this will be discussed in greater detail in one of our upcoming webinars in this series. It is important to note, however, that drugs that cause physical dependence do not necessarily have abuse potential, as is the case with drugs such as SSRIs and SNRIs used in the treatment of major depression. Next slide, please. There are established guidelines available to address abuse potential evaluation. Both the FDA and EMA have issued guidances as seen here on this topic. Next slide, please. In comparing both the FDA and EMA requirements for abuse potential, both agencies require preclinical testing, which includes in vitro testing as well as in vivo preclinical testing, including animal models evaluating abuse potential, reinforcing effects, and physical dependency. These are noted here as Tier 1 and Tier 2 studies. The key difference between the agencies is that the FDA, as well as Health Canada, require clinical evaluation. We will cover these studies in greater detail during this presentation, including any exceptions to these requirements. Next slide, please. Abuse potential evaluation serves as an important guide to determine whether a drug will be considered a narcotic and require scheduling or controlling. In the United States, this process falls under the Controlled Substances Act, where drugs are classified into 5 schedules that are based on their accepted medical use, their relative abuse potential, and the likelihood of causing physical dependence. In Europe, the scheduling of new drugs is performed at the national level. The EU member states classify drugs and precursors according to the UN conventions, and no direct recommendations are made by the EMA. Next slide, please. This table illustrates the 5 classes of scheduling in the United States. As you can see, as you move from Schedule 1 to 5, there is lower abuse potential and less restrictive prescribing restrictions. For example, a Schedule 2 substance, such as opiates, cannot have telephone prescriptions or refills. Schedule 5 substances, however, may be dispensed even without a prescription. Schedule 1 and 2 both contain drugs with the highest amount of abuse potential. The main difference here is that Schedule 1 drugs are not approved for medical use in the United States, and Schedule 2 drugs do have approvals for medical use. Next slide, please. Abuse potential is generally conducted for new drugs when the drug and/or its metabolite affects the central nervous system. If it's chemically or pharmacologically similar to other drugs with known abuse potential, or if it produces psychoactive effects such as sedation, euphoria, amongst others, or if it's formulated with abuse deterrent properties. Abuse deterrent formulations follow another specific guidance that the FDA has issued. In some cases, marketed drugs may require abuse potential evaluation. This has been requested for some drugs that presented unexpected adverse event profiles that indicated abuse potential, or in cases where a new route of administration was being considered that could increase the reinforcing effects of a drug. This generally occurs with routes of administration that increase the rate of exposure and entry into the central nervous system. One example would be introducing an IV formulation for a marketed drug that is normally given orally. Next slide, please. Now we will cover the first tier of studies that are required by both the EMA and FDA, the pharmacology in vitro studies. In vitro studies typically conducted early in the drug development program provide helpful data to evaluate abuse potential. This falls under the realm of chemistry and manufacturing and includes examination of the molecular structure to determine if it's structurally related to drugs with known abuse potential, physiochemical properties including solubility and melting point, factors which are relevant if a drug may be abused by an unintended route of administration such as injection, and a description of the manufacturing process, particularly if the process involves a precursor that is a controlled substance, or if the drug molecule can be converted into a drug with known abuse potential. Next slide, please. The importance of in vitro data is best described by the example of pseudoephedrine. Pseudoephedrine is a sympathomimetic drug of phenethylamine and amphetamine and is the ingredient in the over-the-counter sinus decongestants. Pseudoephedrine achieved notoriety when it became used as an ingredient in the illegal manufacture of methamphetamine. Subsequently, the U.S. passed an act of 2005 to limit purchasing quantities and restrict over-the-counter sales. Next slide, please. In vitro receptor binding studies also play an important role in the determination of abuse potential. Drugs are evaluated for binding capacity at a large number of receptor sites, and some receptor sites are known for their involvement in drug reinforcing effects and abuse potential. This includes binding to known targets such as dopamine, GABA, opioid, cannabinoid, and other receptors. Functional assays help to determine if the binding is agonistic versus antagonistic in nature. A novel drug that binds to these targets will likely need to be investigated more thoroughly for abuse potential. Next slide, please. Now I will turn over the presentation to Andy Mead, who will review the second tier of abuse potential evaluation that are common to both the FDA and EMA, and these include the preclinical behavioral pharmacology and pharmacokinetic studies.
Thank you, Beatrice. So as Beatrice has just said, I want to take sort of the next 10 or 15 minutes to walk you through what's required from a non-clinical or preclinical behavioral pharmacology assessment, when you might consider doing these, but also take the opportunity to try and pull out some key differences across sort of US and EU expectations from a regulatory perspective on these assays. So can I have the next slide, please? So in terms of the timing of these studies, well, it's not set in stone. It tends to be around the end of phase 2, either in readiness for the end of phase 2 meeting or rapidly following. The exact timing tends to be driven by business strategy, but also maybe the level of uncertainty and confidence you have in the strategy that you're taking. In terms of which molecules this is required for, from an FDA perspective, this is a fairly simple assessment in terms of an abuse potential and non-clinical abuse potential assessment is required for all CNS-active molecules. From an EMA perspective, it's a slightly different scenario, which is it tends to be considered for all CNS-active molecules, but they present at least a theoretical option for not conducting some studies for precedented mechanisms. So these can be precedented mechanisms where either the class has already been shown to have an absence of dependence or abuse potential, or the inverse, where the class or the mechanism is associated with a demonstrated or proven presence of abuse potential. So I use the term theoretical option here just to highlight some key caveats to taking the approach of not conducting a non-clinical abuse potential assessment from an EMA perspective. And the next sort of bullets on this slide really talk you through the requirements for not conducting those assays. So following in vitro and in vivo profiling, and when we say in vivo here, we are speaking about a fairly basic sort of CNS assessment, behavioral pharmacology assessment. Further studies investigating dependence might not be necessary when three conditions are met. The first of those is that there's no interaction of relevant concentrations with molecular targets considered relevant for dependence potential, or when the binding to those targets is observed, but you don't see a functional effect associated with it. So these are the targets that Beatrice just introduced in the previous few slides. Also, the situation would be that in vivo investigations do not point towards dependence potential, and a third condition, that the active substance does not have a novel mechanism of action. So the underlining on this slide is my own, just to emphasize, I guess, a key point in the reading of the guidance in this area, which is those terms are fairly subjective in nature in terms of defining what is a relevant concentration, what are the relevant targets, and so on. So, the caution should be taken if you're going to take the approach of not conducting the non-clinical abuse potential assessment from an EU perspective, in terms of whether your assessment of relevance or novel is the same as perhaps the regulators. In addition, they go on to say that if signals pointing towards drug dependence potential would emerge from subsequent studies, either in non-clinical or clinical setting, then you may need to reconsider that decision not to perform the non-clinical assessment. So again, there is a risk there. Fairly late in development, signals may emerge that require you to go back and do the non-clinical assessment. Next slide, please. So when we speak about the non-clinical data, what does that include? And this slide gives a sort of top-level overview of exactly that. And the first two bullets Beatrice has already introduced in terms of the binding profile to targets known to be associated with abuse potential, as well as the functional activity at any of those targets. And so you build from there to develop an understanding of whether any of that in vitro functional activity translates through to the in vivo setting, but also a broader CNS phenotyping assessment, including, for example, locomotor activity. We know that the vast majority, if not all, drugs of abuse will alter locomotor activity in non-clinical species. And then building from there, there are three assays which are somewhat bespoke to the abuse liability assessment. The first of these is the drug discrimination, which asks the question of the animal, does the drug feel like, is it perceived as being similar to a known drug of abuse? The self-administration assay, which asks the question around whether the drug has positive reinforcing properties, and the physical dependence and withdrawal assay, which asks whether a cessation of subchronic dosing with the drug results in the occurrence of negative signs and symptoms that themselves may encourage subsequent drug taking. And of course, all of this data is contextualized in the same way as the vast majority of drug discovery, drug development data with PK and exposure information. Next slide, please. So let me walk you through each of those assays in a little bit more detail, and again, try to pull out some key differences from the regions. So the self-administration assay is sort of exemplified by the schematic on the left of this slide, where you have an animal in an operant chamber or Skinner box, where it has the choice to respond on two levers. And responding on one of those levers activates an infusion pump, which administers a dose of the drug intravenously to the animal. And so you're essentially asking whether the animal is willing to perform that response in order to obtain repeated infusions of the drug and maintain that responding. And the panel on the top right just shows the classical inverted U-shaped dose response that you see for a drug of abuse or a positive reinforcer in this assay. The key aspects of the assay are that the drug is usually administered intravenously, and the primary reason there is simply to allow a rapid temporal association between the response, so in this case the lever press, and the presence of the drug, the administration of the drug. It's an assay which has good concordance with positive subjective drug effects in humans, and we've been able to assess that by looking at over 100 molecules which have been tested in both of those assays. And it also, of course, has excellent face validity. This is an animal that is choosing to self-administer the drug of its own free will, in the same way that we think about humans choosing to take the drug of their own will. Next slide, please. So even though I've introduced it as a single assay, how you run that assay enables you to ask slightly different questions. And that's an important piece of how you design a package using this assay. So the panel on the top left shows responding for 3 opioid analgesics: remifentanil, morphine, which are Schedule II opioids, and tramadol, an atypical Schedule IV opioid analgesic. And you can see that under what we call a fixed ratio schedule, so this is where the animal has to press a fixed number of times for each infusion, in this case 3 times, you can see that remifentanil maintains much higher rates of responding than does morphine or tramadol. And it's tempting to say, therefore, the animal wants remifentanil more than it does morphine or tramadol. But that isn't the case. The reason the animals are taking higher numbers of infusions for remifentanil is simply a reflection of the half-life of remifentanil, which is an ultra short-acting opioid. So this is an assessment in this assay of it's a purely qualitative assessment of whether the drug is or isn't reinforcing. But there are ways that we can tease that apart and start to ask slightly more refined questions about whether one drug is more reinforcing, more rewarding, or is wanted more by the animal than another. And an example of that is on the top right. So this is using the same 3 drugs, actually in this case in the same animals, under what we call a progressive ratio schedule. So instead of needing to press 3 times for each infusion, in this scenario, each time the animal gets an infusion, we increase the number of times it needs to press for the second one. And we continue to do that until the animal stops pressing. So we're really asking the question, how hard are you willing to work in order to obtain an infusion of this drug? And you can see here that in this case, morphine and remifentanil actually maintain very, very similar levels of responding, consistent with the fact that these are both Schedule II drugs. In contrast, tramadol maintains a lower level of responding, again, consistent with a lower level of scheduling. So why have I talked you through this? Well, that comes from the text on the bottom of this slide, which is that there's actually very different perspectives from FDA and EMA when it comes to the use of schedules such as progressive ratios. So from an FDA perspective, final testing should be conducted using a standard fixed ratio schedule, and they discourage the use of the progressive ratio schedules. In contrast, the EMA said that actually when possible, progressive ratio schedules should be used to allow assessment of relative reinforcing efficacy. Next slide, please. So just to move on to the next of these models now, this is the drug discrimination model, which is done in a very similar setting, but actually involves a very different setup within that. So again, it's an operant chamber, and there are 2 response levers, but instead of responding on the lever to obtain an infusion of drug, the animal is responding on the lever to obtain, for example, a food reward. And the animal needs to learn which lever to press. And the way it does that is it uses the discriminative cue, the interoceptive cue, that is induced by a drug of abuse. So before the session, the experimenter doses the rat with either a drug of abuse, or a vehicle. And if it's given the drug, the animal has to press the left lever to get the food reward. If it's given the vehicle, the animal might have to press the right lever to get the food reward. So it's having to sense and show a sense of perception of how it's feeling to solve the task correctly. Over a matter of weeks, animals will typically learn that task fairly robustly. And once they've done that, it gives you an animal in which you can ask an interesting question, which is to come in with a test drug and say, well, if I give you a novel drug, do you perceive it as being similar to the training drug, the drug of abuse, or not? And that's what's depicted on the far right in the data there. So these are animals which were trained up on the CB1 agonist, CP55940. And you can see that actually when you give either that training drug or indeed another CB1 agonist, delta-9 THC, the animals choose the CP55940 lever, the training drug lever, showing that they see it as being similar as a training drug. In contrast, another drug of abuse, in this case cocaine, doesn't show the same response. And that's an important point about this assay. This isn't about whether or not a drug has abuse potential, it's about whether it is similar to the draining drug. So cocaine, of course, is completely recognized as having clear abuse potential, but of a very different mechanism, a different pharmacology to the draining drug in this assay. In terms of EMA and FDA guidance, generally speaking, they're fairly well aligned in terms of expectations around drug discrimination. They both emphasize the choice of training drug as a key variable. And I guess if there's one difference between them, it is that the EMA suggests that you might look at more than one comparator for novel mechanisms. That is, use the drug discrimination model as a way of identifying appropriate comparators for maybe some of your other non-clinical or even a human abuse potential assessment. Next slide, please. So the final of the three models I was going to speak to is the physical dependence on withdrawal. So again, as a reminder, this is an assessment of whether cessation of subchronic dosing results in the development of a withdrawal syndrome that itself might then encourage sustained drug taking. So the two data panels on this graph show rats that have been given 14 days administration of pentobarbital and then abruptly ceasing that administration. And you can see that during the withdrawal phase, these animals are, first of all, more difficult to remove from the cage, so a basic behavioural clinical observation. But on the right, we've actually got a different assessment, which is it's a break point reached for food. So this is a progressive ratio for food, which is often used in animals as a measure of motivation or an indication of an anhedonic state. And you can see there a very sensitive measure of withdrawal with the pentobarbital route. So again, there's broadly good alignment across EMA and FDA regarding expectations for this model. Both of them emphasize a number of key points, one of which I've just spoken about, which is the importance of choosing appropriate endpoints for the drug class. So every drug class has a very distinct withdrawal syndrome. We see that clinically as well as non-clinically. And so it's critically important that we choose the appropriate endpoints for the class of drug that's being assessed. They both acknowledge that both spontaneous and precipitated withdrawal are acceptable. So spontaneous withdrawal refers to simply cessation of dosing one day. Precipitated withdrawal is where, for example, if your test drug is an agonist and you're dosing that for say 14 or 28 days, you then administer the antagonist on top of the agonist on the final day to precipitate potentially a more severe withdrawal state. They both emphasize that it's important to consider the sufficient duration of dosing, but don't necessarily go into details about exactly what that is. And interestingly, they both comment that the assessment of tolerance is not required. So with drugs that induce withdrawal, it's not unusual to also see tolerance during the dosing of the drug, but it's not a necessary link, and therefore they highlight that that in itself is not a key factor. Next slide, please. So just to finish from me, just some additional, I guess, generic differences across the regions that are worth flagging. So generally speaking, the expectations are fairly similar, but there are some methodological differences regarding study requirement design. And one of the key ones there relates to dose selection. So when we think about the high dose selection in these studies, the FDA suggests that we focus on doses that produce that are benchmarked versus Cmax from your highest proposed therapeutic dose. And typically that is sort of up to 2 to 3-fold times higher than that. So we're talking about pharmacological doses here. The one exception being self-administration, where we come a little bit lower, but it's still pharmacological doses. EMA, on the other hand, suggests that actually you could push up to the MTD, the maximum tolerated dose, if this doesn't interfere substantially with the behavior, so up to potentially more toxicological doses. And there's a similar distinction on the low dose selection. So the FDA suggests, again, informally based on the relationship to clinical Cmax, the EMA suggests actually you should be starting from threshold doses, those that show no behavioral effects. So being aware of these is absolutely key in terms of how you design your studies to meet the objectives from both agencies. Both of them suggest that positive control is required, but again, there is an exception or a potential exception from a European perspective, which is that if there's sufficient documented data to show the validity of the assay, perhaps you don't need a positive control in your studies. And again, it's worth flagging that the term sufficient is, of course, a very subjective term. So as a take-home message, studies can usually be designed to meet the requirements of both EMA and FDA, as long as there's an appreciation of some of these subtle differences across the guidance documents. And I'll finish just by saying that both of them are completely aligned from the use of the rat as a preferred species unless considered scientifically appropriate. So of course one shouldn't use the rat if the pharmacology of the drug, for example, is not expressed in the same way in the rat. And that's a position that is reinforced in ICH-M3 as well. So on that, I'll hand back to Beatrice to take you through the human abuse potential assessment.
Thank you, Andy, for that nice overview. We will now cover the third tier of abuse potential studies that are required by the FDA specifically, and these include the human abuse potential study and the clinical evaluation. Next slide, please. Abuse potential is evaluated throughout the clinical drug development program, as you're seeing through in this presentation. The dedicated human abuse potential study, however, is generally conducted a little bit later and after the end of phase 2 meeting. This meeting serves as a critical regulatory milestone with the FDA to review the psychoactive drug effects, if there are any signals of abuse potential, and to establish whether further testing of abuse potential is required, or if, in some cases, if the requirements may be waived. And this will be entirely dependent on the data collected and whether or not you have certain adverse events that we'll discuss shortly that are presenting themselves that would warrant further investigation. The human abuse potential study is conducted in this phase as at this stage is sufficient enough data has been collected to understand the pharmacological drug profile, the therapeutic and maximum tolerated dose levels have been identified, and this information is required to design a robust human abuse potential study. So for these reasons, they're conducted a little later in the program. Sponsors who prepare early and evaluate their data for abuse potential are in far better positions to have comprehensive dialogue with the FDA concerning the need to conduct these studies at the end of phase 2 meetings. So it's really about keeping an eye out on all of the data that you're collecting from the very, very beginning. Next slide, please. Data relevant to abuse potential is captured in every single clinical trial in the form of adverse events. There are key adverse events of interest that are identified in the FDA guidance that are associated with abuse potential. Such events reflect psychoactive effects, and these include things like euphoria, feeling drunk, somnolence, amongst several other important adverse events. It's important to monitor these events and in some cases capture more details in the forms of narratives, as some of these events may be subjective in nature and they may be subject to interpretation. So whenever you can, the verbatim terms from the patients can often be very helpful in elucidating where the elation of mood is coming from, for example. Other endpoints of interest include incidences of aberrant pill counts, which could imply either overconsumption or diversion. Hence, proper data collection at the time of study conduct is critical to distinguish between accidental versus intentional overconsumption, for example. In addition, any reports of overdose and details of the event are of importance to include. So ensuring appropriate data capture and monitoring is key to a comprehensive abuse potential evaluation. Next slide, please. The dedicated human abuse potential study serves to evaluate the likability or attractiveness of a drug for abuse in a face-valid population. These studies are single-center, single-dose, double-blind, crossover design studies. The study population comprises typically of normal, healthy males and females, generally between the ages of 18 and 55 years of age. And these subjects are not physically dependent on drugs or alcohol. The subjects will be selected based on their past recreational drug use history with the class of drugs selected as the positive control in a given study. These types of studies include both a placebo and an active control. The active control is a scheduled drug with known abuse potential and generally falls into the classes of either opioid stimulants, sedative hypnotics, cannabinoids, or hallucinogens. Based on the pharmacology of the investigational drug, the active control is selected based on similar properties of psychoactive effects. In some cases, more than one active control may be selected, and we also take into account the indication, the intended indication when selecting appropriate controls. Prior to the main study, a pharmacological challenge is conducted to confirm subject eligibility. This enables us to confirm that they're not physically dependent on a drug. So for example, with recreational opioid user, a naloxone challenge is done to ensure that they're not physically dependent on opioids. But also importantly, we need to confirm that they are sensitive to the active control and can distinguish it from placebo on the measure of drug liking and other subjective drug effect endpoints. This confirms study validity. Next slide, please. In some cases, controlled and ventilated chambers are required for substances that are delivered as aerosols or through inhalation or smoking. This feature ensures that exposure is contained from staff and other subjects. Our clinic sites are customized and include these types of chambers for various different types of administrations and formulations. Next slide, please. One of the key primary endpoints in a human abuse potential study is drug liking. The picture here shows this measure as it's presented on a 0 to 100 point visual analog scale. The question simply asks, at this moment, my liking for this drug is, and the subjects select the degree to which they like the drug in either direction on the scale. To the right is increased drug liking, the middle of the scale is neutral, neither like nor dislike, and the left side is increased drug disliking, so they do not like the drug. This is what we call a bipolar scale because there are two different opposing anchors on either side. Additional endpoints in these studies include other evaluations of subjective drug effects, which are customized to the investigational drug, and these will also include measures such as the willingness to take the drug again. The measures of psychomotor skill and cognition are also included to determine if a drug exerts any impairing effects. These measures are usually always determined on tablets, but can also be administered as paper pencil tests. I will say that measures of cognition and psychomotor skill are often beneficial to include even in early studies in phase 1, such as the first in human study, to determine whether or not there are any signals that may warrant further investigation down your clinical development pathway. And that'll be a focus of a topic in a future session. Next slide, please. A recent example of a newly marketed drug that underwent abuse potential evaluation is Epidiolex, which is cannabidiol, an ingredient derived from marijuana. Epidiolex was approved for the treatment of seizures associated with Lennox-Gastaut syndrome or Dravet syndrome in patients 2 years of age or older. The figure shown here presents the primary measure, which is drug liking. On the y-axis is 0 is strong disliking, 50 is neutral, and 100 is strong liking. As you can see, the active controls alprazolam in the gray line and both doses of the active control dronabinol, the pink and red lines, showed high levels of drug liking over time. In contrast, the 3 doses of the cannabidiol, which is the Epidiolex, shown in the 3 shades of blue, and the placebo, which is indicated in the orange line, were very similar and at the neutral point for drug liking. Next slide, please. Based on the collective data obtained for Epidiolex, which of course would have included all the preclinical and clinical data, this drug received an unscheduled status. Data to support that Epidiolex does not show signs of abuse potential is provided in section 9 of the label. Such labeling serves to differentiate a drug from other drugs with similar indications for seizures that are scheduled, for example, benzodiazepines, which are generally falling into the Schedule 4 class. Some sponsors choose to conduct these types of studies to strategically differentiate from other in-class drugs that may be associated with higher abuse potential. So you can certainly see a case for this when you're developing drugs that are targeted for epilepsy, seizures, sleep, as well as pain, often a strategic maneuver. Next slide, please. At the time of an NDA submission, there's a list of data that is collectively reviewed to determine the abuse potential and need for drug scheduling. The review process is conducted across several agencies during the NDA review, and it includes the FDA, which includes also the Division of the Controlled Substances staff, the Department of Health and Human Services, NIDA, which is part of the NIH, and ultimately the scheduling determination is actually made by the DEA, the Drug Enforcement Agency. All of the preclinical and clinical data listed here is reviewed in the form of an 8-factor analysis. Next slide, please. The 8-factor analysis is conducted internally by the FDA as a method for their review. The 8 factors cover the information related to the drug's abuse potential and includes any post-marketing data that may be relevant and a discussion of public health and safety. As part of the NDA submission, the sponsor is required to propose a scheduling status. And oftentimes conducting your own 8-factor analysis and walking through what the FDA and these agencies will be looking at as part of your NDA submission can help organize your data and provide a strong justification for why you feel your drug should be scheduled or unscheduled, and if scheduled, what schedule it should fall under. We often consult with clients and will write 8-factor analyses to help sponsors at the time of the submission and other critical regulatory milestones. Once a drug is scheduled, a drug generally remains in that schedule. There are some few exceptions of drugs that have received status of a therapeutic indication and have moved from Schedule 1 to 2 because of the approved therapeutic use. There are also some instances where the agency has gotten more conservative and scheduled to a more conservative schedule. But generally, it usually tends to stick. So the best strategy is to provide the most comprehensive data to obtain an appropriate schedule at the time of the NDA review. Since schedule can impact the prescribing practices of a drug, it's an important component in the drug development and review process, which starts from day one and continues with you throughout the development cycle. Next slide, please. So in summary, CNS drugs require unique risk-benefit evaluations that occur from the start of the drug development and continue throughout the entire drug development process. Including assessments early in drug development and examining your data along the way helps to identify the degree of testing that may be required for your program. And it also provides important safety information for the design of phase 2 and 3 studies. Understanding the preclinical and clinical requirements can avoid costly delays in drug approvals for both FDA and EMA submissions. Finally, the strategic review and compilation of this data can help support your scheduling status recommendations. Andy and I are experts in this area and can help you navigate these critical steps throughout your entire drug development process. This concludes the presentation portion of today's meeting. We thank you for joining and are happy to answer any questions from our audience at this point. So Andy, if you could unmute yourself, I see we've got a number of questions that have come in. If you have other questions that occur to you after the meeting, or if there are things that you have very specific to your program that you wish to directly communicate with us, we will also leave our contact information here for you to be able to reach us with additional questions you might have. So why don't we go ahead, Andy, and get started. We've got a question that came in regarding in vitro receptor binding studies. Should metabolites be evaluated if they are known to be major metabolites that are brain penetrant? The answer to this, to the receptor binding, is yes, that those should be. And Andy, that's also tied into the preclinical studies, and the follow-on question is how to deal with a compound that does not produce the same metabolites in the preclinical species as in human if those metabolites are known to be CNS penetrant and have activity in vitro functional assays. So there's one where you have a discrepancy between human and animal metabolites. So for the answer for the first question, yes, we are looking at metabolites and receptor binding. Andy, would you like to tackle the preclinical question?
Yes, and it's a good question that comes up reasonably frequently, actually, and there's no short answer to it. The first question is, can you consider the non-rodent as your preclinical species? Is this truly a human-specific metabolite, or is it that you can get around this with the non-human primate? In which case, you may well consider a switch of species in your non-clinical studies. If that doesn't help you, if we're talking about a truly unique human metabolite that isn't produced in either rat or non-human primate, then you can consider studies to assess the metabolite directly from an abuse potential perspective. We don't often get into that situation, but it can happen. But I think the first thing to do, as was alluded by these questions, is definition of metabolites, which need to be assessed as well. So is it a major metabolite? I mean, FDA use the same as MIST in terms of 10% of parent. So is it CNS-active? Does it bind, as we've already alluded to any targets of concern? And then the final one is percentage of parent. But there are a number of ways of addressing that. There's no single answer to that question.
Mhm. All right, thank you, Andy. Our next question is, again, around the non-clinical studies. If you plan your non-clinical studies to be fully performed by end of phase 2, so before end of phase 2, how do you deal with the common uncertainties of the final therapeutic dose? This will only be known towards the very end of phase 2. So Andy, what do you do in those types of situations? Do sponsors defer to conducting them at the end of phase 2 after they've had their end of phase 2 meeting? Or are you seeing a lot of sponsors that come to you before that time frame, and how do they deal with the therapeutic dose?
And clearly this is an important question, because I see at least 2 people have asked a very, very similar question. No, it's important to address this. And I think that the key way to think about this is the risk is purely on the sponsor's lap if they do the studies earlier than end of phase 2 or earlier than they have selected their phase 3 dose. But I think the one thing I'd mention is that for the non-clinical studies, it's not unusual for us to test a reasonably broad dose range. So if you take, say, 4 doses in the self-administration or the drug discrimination assay at half-log units, you've got a 30-fold range of doses. So you can design those studies in a way that gives you a little bit of leeway should your phase 3 dose be higher than you expect it's going to be. So that's the mitigation against getting into some surprises around your final phase 3 dose selection, and covering yourself and not having to repeat those studies.
Okay, thank you, Andy. And next one, Andy, for you, is the progressive ratio necessarily better than the fixed ratio if the half-lives are the same?
Short answer is better in that it assesses a different question. It's still appropriate. It still assesses a different question. You can have 2 drugs with the same half-life, but vastly different reinforcing efficacies, which one would expect would translate into different breakpoints or maximum response rates in a progressive ratio schedule. So yes, it is. It maintains the advantages it provides, regardless of the half-life of the drugs that you're testing.
Okay, great.
I should probably add, within reason, there's sort of one exception there. If you're talking about a molecule with an extremely long half-life, then there are challenges in running both those models, whether it's the fixed ratio or the progressive ratio schedule. But that's a slightly separate discussion.
Okay, and then for in vivo studies, Andy, which species could be considered if the rat is an inappropriate species?
All the questions are coming through for me. So generally speaking, non-human primate, and then within that, it really does depend on the mechanism that you're looking at. So to give you an example of that, for cannabinoids, it's not unusual to use the squirrel monkey for an assessment of abuse potential for self-administration assays, I should say. There's an extensive history of the use of the particular species in there, but it's always non-human primate or rat. Or potentially mouse, although we don't see that very often, I would add.
And then the next question, how can you finish the study by the end of phase 2 meeting and pose questions when you need the exposure Cmax from phase 2 to design these studies? So part of it, the design and the end of phase 2 meeting, there's a lot of other data that's going to be collected at that time of the end of phase 2 meeting. So even if you don't start, let's say in a scenario where you don't start your preclinical models that Andy reviewed, as well as the human abuse potential study, they're looking at some of the other in vitro data. They may be looking at other animal pharmacology studies that have been done. They'll be looking at primarily adverse event data collected up until your end of phase 2 meeting that may be indicative of these signals. So there are a lot of other types of data that can lead to an informed dialogue with the FDA, even if you haven't started the preclinical studies or you've been waiting for the FDA to give you guidance. In some cases, when sponsors don't have any signals and they've included a lot of, oftentimes we have very complex first-in-human studies where sponsors are including some type of psychoactive measures, or they're looking at cognition and other types of indicators to show how that CNS effect is warranted. So there are other pharmacology studies that become very informative in the discussion. And in some cases where there's a blank slate for signal, it's possible to waive some of these studies or all of these studies if that's the case. However, it is the timing of it really will be specific to your program, and certainly you do have other reliable data for an end of phase 2 meeting, even if these are deferred to later in your development program. Andy, I don't know if you had any other thoughts on that.
No, I think this is a similar question to the one I answered earlier,
Mm-hmm.
so nothing additional to add.
Mm-hmm. And Andy, I think that the next one is for you again. Would you recommend to perform a withdrawal study in rats at the same time as the drug discrimination and the self-administration study, or rather wait until you know whether you have to do a human study?
So I think the timing of the withdrawal study depends, as is so often the case. It really depends on your development program and your intentions of maybe engaging with the agencies around need for subsequent studies. But I guess what I would add is I don't see that the need for a human abuse potential study necessarily influences the need for the non-clinical withdrawal study. That would be an expectation for a CNS-active molecule, regardless of whether you need to do a human abuse potential study.
Okay. The next question is, would it be feasible to add an extra arm in a phase 1 SAD study to study human abuse potential in comparison to naive human volunteers? Now, we get this question quite a bit, and I oftentimes will stop sponsors and say, be careful what you may be asking. The drug liking and a lot of the primary measure, which is drug liking, and some of the other subjective drug effects are very much tied into the recreational drug using experience. When you're using these in naive human healthy volunteers, it's not a valid question for that population, particularly because if they're not abusing drugs or don't feel those types of positive reinforcing effects, you introduce a lot of variability into the study. So it's oftentimes far more effective to use the questionnaires in the correct study population, in the human abuse potential study. And I can tell you that even having conducted human abuse potential studies for many, many, many years, the variability even within the recreational drug users is quite variable, which is why we use that qualification or that drug discrimination phase. So you would simply introduce far more variability, generate potential signals in healthy naives that would then require a lot of work to have to explain to the agency. So when you're dealing with SAD studies or early phase studies in healthy volunteers, other questions perhaps could be appropriate to evaluate, perhaps if they're feeling any kinds of pleasant effects or good effects, or there's certain questions that may be more appropriate and are easier to understand in that population. But certain things like for certain you can include measures of cognition and psychomotor skill and other types of measures that are appropriate in that population that can give you insight into the drug pharmacology and its behavior and the CNS effects it poses. But that's a great question, and that's one that comes around quite often. The next question is, could you comment on the regulatory expectation of the type of preclinical studies needed, if at all, if one wants to combine a CV drug with a Schedule 5 drug with a Schedule 3 drug? So this is a drug combination question.
I think this is a really interesting question. And actually, I don't have a really good answer for this, because we don't often get into the situations of combinations, and often when we do, we get into some interesting challenges about exactly what is required. I think one comment I would say is if we're talking about a marketed, and presumably we are, that we've got a Schedule 5 and a Schedule 3 drug, well, those drug substances remain scheduled as Schedule 5 and Schedule 3 if we're simply talking about a combination of two already marketed products. So it doesn't necessarily change anything. Would there be expectations around additional preclinical studies of the combination themselves? I genuinely don't know the answer to that question. It's a really interesting one.
Mm-hmm. Okay. And the next question is, could you please elaborate on when and how the waiver can be considered for human clinical abuse trials based on the FDA regulatory perspectives? The time to do that and the time to really make that evaluation is at the end of phase 2 meeting. So, and again, this is very much based on strategy. If this is one of your intent, then collecting and organizing all of the data that you've collected up until that point, and perhaps proactively, perhaps even looking at some of the preclinical models, it can be a provide substantial enough data to determine whether or not there's a signal. And usually you need a pretty clean slate in terms of not having these signals in the adverse event data and in other places to have the FDA say, well, you know what, there's really no signal. So it's based on the mechanism of action, it's based on the receptor binding, it's based on all the collated data that's up to that point. So it certainly can happen, but if there are signals, then brace yourselves, because there's likely that the full investigation will be required. And then I think that also ties into a question with whether or not one can just skip over to the human abuse potential study and waive the preclinical, or just do the preclinical and not have to do the human abuse potential study. From my experience, when the human abuse potential study is needed and when the FDA is asking for that, it's usually the full package that's required for the abuse potential evaluation, because it becomes a regulatory checkbox that these were done. Andy, have you encountered situations in which one or the other was waived?
So no, I generally agree, which is if this is a CNS-active molecule that warrants an assessment, then you would certainly do the non-clinical. I have seen situations where, having come out of the non-clinical, when you have a completely clean package, that of course a decision has been made not to pursue the need for a human abuse liability study. But that's a scientific argument rather than simply getting away without doing one of the studies.
Mm-hmm. Thank you. Andy, the next question directed to you, is conditioned place preference a good indicator for non-addiction?
So I hope they don't mind. I'm going to reword this question slightly, and conditioned place preference is a useful alternative assessment of drug reward. So an alternative, probably in the majority of cases for the intravenous self-administration. Like any behavioral pharmacology study, it comes with its pros and its cons, and it is useful in instances, for example, where you really couldn't run an intravenous self-administration study. Perhaps you can't, there's no intravenous formulation that would allow you to test the exposures you need to do, and I think it's a useful substitute in such cases. I think the IVSA model is by far and away the default choice, and that's supported by the regulatory position as well. So yes, it's an assay that has value, but it's an assay that has value in situations where you're trying to address questions that can't be addressed from the core package.
Mm-hmm. Mm-hmm. Thank you, Andy. The next question, how are compounds handled which are potentially converted into psychoactive metabolites or even a known controlled substance? And I think in this case, when you're dealing with something that is converted to something with known abuse potential or psychoactive, the abuse potential generally in those cases would need to be evaluated according to the guidance. If it's a psychoactive metabolite, again, it will depend on whether or not it produces a lot of these signals as to whether or not the full abuse potential evaluation is conducted. So these are always case-by-case basis based on the available data and the pharmacology. The next question, can one leverage other clinical data to forgo a dedicated human abuse liability study? Yes, I think we've covered that a little bit earlier about the other types of data generally that goes into the end of phase 2 meeting at that critical discussion. So if the FDA requires that you do a human abuse potential study, you're going to need to do that as part of the final NDA submission. But at that end of phase 2 meeting, you can certainly look at other clinical data to make that decision with the FDA. And certainly if it's needed and then you're submitting your NDA, you'll be looking at all of that clinical data as part of the justification for scheduling status. Next question, would a new dosage form with the same route of administration of an approved drug, example, a new tablet, when an oral solution is approved, require abuse potential studies? And will in vitro and animal studies be required? Well, it depends. The only case that comes to mind where we had differential scheduling based on an oral formulation is really the syndrostrobinol. And one formulation was it was approved as Schedule 2, whereas some of the other same molecules were given a Schedule 3 status. And the difference was that the one formulation was in liquid and contained alcohol. And because of that, there was more kind of sensitivity about that. But if you're going from an oral solution to a tablet, unless you have a marked change in pharmacokinetics, then I wouldn't imagine that additional testing would be required. Andy, any of your thoughts from your experience?
I was going to give a very, very similar answer. I think from an in vitro perspective, I struggle to see a scenario where you would have benefit in testing there. And actually, when the animal studies are considered, it would only be where you had, for example, a marked change in PK. And you could argue that maybe the physical dependence and withdrawal study, maybe there's a reduced propensity for withdrawal with an extended sort of flatter PK profile, for example, and that may actually be beneficial to the sponsor to do so. But no, I think as a general rule of thumb, unlikely.
Yeah. And again, a question came up on the bypassing. If clinical studies are required, can you bypass the need for the animal studies and go straight to the clinical abuse potential evaluation? Generally, no, because it's a regulatory checkbox. If that evaluation is needed, you're going to need the full package to support that submission. Andy, any exceptions to that, or has that been the case? I think we confirmed that earlier. Okay.
No, nothing there.
If there are no clinical indicators of abuse potential, assuming all the right evaluations were performed and no non-clinical abuse liabilities were performed, but then abuse liability suggestive AEs show up in phase 2-3, I expect you would need to evaluate abuse liability in a clinical study. You wouldn't need to go back and do non-clinical assessments, would you? Well, yeah, you likely would. I think what you have to keep in mind is that when the FDA expects an abuse potential evaluation, they're expecting it to be done by the guidance. So that includes the preclinical and the clinical. And sometimes these can show up late. And we've had, in fact, situations like we talked about in the presentation where it happens in post-marketing. Sometimes it, depending if you have a 505(b)(2) submission, that is a little bit different. But when it's hinged on a scheduling decision, they may very well go back. And we've had situations where sponsors had to go back and perform some of this data before the NDA would get accepted. And so therefore, it's always important to be very vigilant with that data and look at that. Unlikely that it would just show up in phase 3 and it never showed up earlier. That's kind of an unlikely scenario, particularly because you're starting to do larger and larger testing in phase 2. But it could happen, and if there's a signal that requires it, it would require the full package. Next question, what is the rate of false positive and false negatives in the preclinical procedures? Andy, that one's for you.
Yeah, and it's a difficult one to answer in terms of quantitative on this call, but actually it's a topic that we've published on in the past of looking at exactly that question. Okay, how often do we get misleading data, whether that's misleading in terms of the prediction of the human abuse liability or misleading in terms of the final scheduling decision from our non-clinical assessments? And of course you do see them. There's very few non-clinical preclinical procedures that don't have false positives and false negatives. The key thing here is to sort of design the studies in the smart way. So some of those false negatives, I think, can be attributed to suboptimal experimental design. But actually, maybe the bigger picture is the right answer to this one, which is this feeds into a few things you've said, Beatrice, a number of times, that when an abuse liability evaluation is required, tends to be the totality of the data that is required, such that if you were getting a non-clinical false positive or false negative, that's hopefully something that would be revealed by the clinical data, the human abuse liability study, and etc. That's why it's an integrated assessment to mitigate against those risks.
Right. Yeah, so next question, and we'll try and see what we can answer here in the next 2 minutes. If a drug going through a 505(b)(2) pathway is being tested at a lower dose than would be necessary to induce drug liking, could you redo drug liking studies in order to get differential scheduling, or is the scheduling based on the underlying drug substance, not the drug product? Yes, it's based on the drug substance. So it's understood that you could end up just manufacturing lower doses. However, anybody can take a full bottle and escalate. And oftentimes the concern is that recreational drug users or drug abusers are not looking at the product labeling instructions when they take their drugs. They'll take as many as they need to get the effect it produces. So it's based on that compound. And that's for these reasons, the human abuse potential studies are conducted at minimally effective, therapeutic, and supratherapeutic doses. So they're always looking at a range even beyond your therapeutic doses. They will include supratherapeutic doses to see what dose escalation may look like. Next question is, what's the best approach for a drug with a novel mechanism of action? Well, more testing. I mean, the more you understand the mechanism of action, the more you understand what it's binding to, and really thoroughly looking at and keeping a very good close eye on the any behavioral pharmacology preclinical studies that you're looking at, as well as the adverse events. As soon as you start looking at that and characterizing that early in your phase 1 first in human studies, it starts to give you a very good indication of how the drug is behaving. So in these cases, and oftentimes if there is a need for a human abuse potential study, a lot of the data that you'll collect will be very informative in understanding what is the drug doing, what is it indicated for, how does it, what kind of psychoactive effects does it produce, and what would be the best comparators for that drug. But I'm afraid we have reached the end of our hour, and there are some more questions that unfortunately we will not have time to get to, but I would encourage the audience to please feel free to contact us and send us an email with your questions. If there are specific things that you'd like to get into about your program, we can certainly put a CDA in place if we don't already have one, and help you out and answer some very specific questions that you may have pertaining to your program. But I would like to thank all of you for joining today and participating. Great questions. Thank you very much. And we're happy to take them offline for further discussions. And thank you, Andy, and the organizers for today's meeting.
Thanks, Beatrice, and I'd just like to echo those thanks as well. Thanks for attending today, and thank you for all the questions. It's been fantastic. Stimulated some good discussion.